Welcome everyone to our Research Connect series. I'm Karina Prévost, a CDTRP PFD partner. I will be facilitating today's webinar. I've been a patient partner for 8 years, and 8.5 years ago, I received a double lung transplant due to cystic fibrosis. First, I would like to acknowledge that the CDTRP is hosted by the University of Alberta, which is located on Treaty 6 territory, a traditional gathering place for diverse Indigenous people, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dini, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continues to influence our vibrant community. We're honored to be uninvited guests on this land and appreciate the ability to work, rest, and play while we are here and leaving the territory as it was when we arrived. I encourage you to write your land acknowledgement in the chat box. We would also like to acknowledge Astellas, Takeda, and AstraZeneca, without whose generous support we would not be able to host these sessions. The CDTRP also relies on the generous support of our community. Your gift ensures we can focus on delivering our mission of supporting excellence in donation and transplantation research. Before I introduce the speaker, I have a few housekeeping notes. Today's session is being recorded and will be posted on the CDTRP website and YouTube channel. During the presentation, we encourage you to keep your cameras on, but we kindly request that you keep your mics off. And if you have a question or comment, you can raise your hand with a raise hand button, and we will get back to you. I'm pleased to be introducing today our presenter, Dr. Mingyao Liu. Dr. Liu is a senior scientist and head of the Respiratory and Critical Care Research Group at Toronto General Hospital Research Institute. He's also the director of the Institute of Medical Science and a professor of surgery, medicine, and physiology at the Temerty Faculty of Medicine at the University of Toronto. He has devoted his research to acute lung injury and lung transplantation. As a co-founder of the Latner Labs, Dr. Liu has teamed up with his colleague, Dr. Shaf Keshavjee, to develop an ambitious lung transplantation research goal. Currently, he focuses on cellular and molecular mechanisms of ischemia-reperfusion injury in lung transplants and developing new lung preservation solutions, EVLP perfusates, and techniques. He has received a number of research awards, including Queen Elizabeth II Diamond Jubilee Medal. We are all very excited for your presentation. I now hand it over to you, Dr. Liu.
Thank you very much. Thank you for the nice introduction. And also it's a great honor to present my research work to this group. It's an excellent group. And let me try to share my screen first. Can you see my screen now?
Yes. Can you go full presentation mode?
Yeah, is that presentation mode?
Yes, this is perfect.
Wonderful, wonderful. Thank you. So the title of my presentation will be Omics and Ischemia-reperfusion Injury in Lung Transplantation. So as Karina introduced, I'm from Toronto, and with my lovely research teams in the Toronto Lung Transplant Program. I think this year is special because 40 years ago, Dr. Joe Cooper performed the first successful single lung transplantation in Toronto. And 3 years later, the first double lung transplantation was also performed here in Toronto. So now lung transplantation become a very efficient therapeutic modality for patients with end-stage disease, and more than 67,000 lung has been transplanted. However, compared with other organ transplant, the median survival of lung transplant is the shortest. It's only 6.5 years. And we are facing many challenges for the lung transplantation. So the big challenge is the shortage of the donor lung. So that's why we have this CDTRP programs to promote our organ donation. And also the second major challenge is specific for the lung. It's a utilization rate. It's the lowest one. It's only about 20% of organ donated will be transplanted, which is much lower than other solid organ transplantation. So to solve this problem, so in addition to the classical DBD transplantation donation after brain death, then we also try to use a DCD lung, which is donation after circulatory death. So it's associated with a lot of challenge on its own. So another major limitation for the lung low utilization rate is because of the fear of ischemia-reperfusion injury. So to solve this problem, about 10 years ago, we developed a technology called EVLP, ex vivo lung perfusion. With this technology, we can assess the marginal donor lung, then to make sure the lung meets the standard before transplantation. So the technology is basically put the lung into a dome like this one. The designing of the dome is inspired by the Sky Dome in Toronto. So the donor lung put in the plastic dome, ventilated, perfused at the body temperature, so which allows us to properly assess the function before utilization. So by using this technology, each year, an increasing number of transplant donor lung has been used, a marginal donor lung has been used. So far, over 800 lung have been processed or assessed by this technology called EVLP, and approximately 70% of these lungs has been used for transplantation. And also we have a recent data to show the clinical outcome for those EVLP lung is equivalent to the regular lung transplantation. And also socioeconomically, from socioeconomic point of view, this technology saves a lot of money from the taxpayers. So it's a very successful technology. And also this technology provide an opportunity for us to do a lung repair. So this has dramatically changed the way we manage the donor transplantation. For example, before the organ harvest, there's a donor management, and then we also have a technology to improve the donor organ preservation, and then the suitable lung after transplantation and will receive the post-transplantation care. And with the EVLP technology, the marginal lung, the lung which the clinician not felt comfortable to use, can be assessed by the EVLP. And if it's suitable, then we can do transplantation. If it's not, with some certain injury, then we can do organ repair, reconditioning, then do a reassessment, then eventually lead to more organ be transplanted. Only the lung with severe injury will be declined. So the development of the EVLP technology really give us a more option and more freedom to solve the ischemia-reperfusion injury. So as a basic scientist in the team, my job is try to develop a drug discovery delivery pipeline for the lung transplantation. So we have the bioinformatics study and to identify potential biomarker and therapeutic target. Then we also develop cell culture models then to screening the drugs, screening the mechanisms before using animals. So in this way, we can have a relatively high throughput screening, and this can save many animals' life before we using a final selected strategy for in vivo test. Then we also have a small animal model include mouse lung transplantation, rat lung transplantation, rat lung EVLP. Then we also have a large animal model include the pig EVLP and the pig lung transplant model, which is the final preclinical trial before we translate our study towards the clinical work. And also clinically, we also can use the donor lung declined from the clinical transplantation program to test the drug even before clinical trial. So to test the unhuman before unhuman. So this is one of the advantage we have. Then we also have a drug formulation technology in the team to do a nano formulation for selected drug. But today, we are only focused on the bioinformatics study, talk about the so-called omics. So what is omics? So omics can be considered as a technology. Basically, you can study one particular category of molecule all at once. For example, for the DNA, you can use the genomics to study the DNA information. For RNA, especially messenger RNA, then you can do a study called transcriptomics. For protein, then you can try to mapping all the proteins in the biological sample, and this is called proteomics. Then for biochemicals, small molecules, then it's called metabolomics. So far, in our team, we have used the transcriptomics, proteomics, and the metabolomics to advance our work to identify the potential therapeutic target and the biomarker. So we also did a literature review recently, try to understand how other teams are doing that in the lung transplantation. Then we are very happy to report that this omics technology have been used in cell culture models, in animal models, include mice, rats, and pigs, and also, of course, used on the human samples. So I will mainly focus on the human samples to tell you guys what we found from the clinical setting with this powerful technology. So before I'm going to the main topics, I will quickly report what we did for the proteomics and the metabolomics. So about 10 years ago, we did a very preliminary work with a bronchial alveolar lavage fluid from the patient who received the lung transplantation. Then we mapping all the protein signatures from that samples. Then through this, we defined a group of molecule which may be related to inflammation. So then we further using a regular technology to verify then the alveolar neutrophil enzymes and also the pantraxin-3 called PTX3 as a biomarker related to a chronic lung allograft dysfunction. And we also found alarmins, which is a group of small molecule, which you also call S100 proteins. They are highly relevant to the development of the CLAD or chronic lung allograft dysfunction. Then in terms of metabolomic study, we used the EVLP perfusate as a sample. Then we mapping all the small molecules in the EVLP perfusate. Then we reported some potential metabolomic biomarkers. But more importantly, by using the bioinformatic tools, we identified two major challenges in the current clinical EVLP. One is the lack of nutrient in the current EVLP perfusate. The current EVLP using a solution called a STEAM solution, which only contains glucose as a major source of energy, and we found this is not enough. The second is the challenge we found from the current EVLP is the accumulation of metabolic bioproducts. So with those problem, the EVLP can be used clinically only for a very short period of time. Usually it's 4 to 6 hours. So if we want to to further improve the function of the EVLP, then we needed to modify that. So subsequently, we tried to using a enriched nutrient in the EVLP perfusate then to improve the EVLP performance. So we have some recent success with that. So this just give you an example by using a proteomics as a guideline to discover biomarker, use the metabolomics as a guideline to develop a strategy which can improve the EVLP performance. But this is not what I really wanted to talk. What I wanted to focus on is the omics in the RR ischemia-reperfusion injury in the human lung transplant. So the first study we did was conducted by Dr. Christina Basu as a PhD bioinformatician. And she actually applied a small funding from the CDTRP, and we received $30,000 support from this program, which is the seed money, which triggered all the subsequent studies. So I want to take this opportunity to really thank the CDTRP to support our research and make our research successful. So what the question Christina asked in her initial study is try to understand if the DBD and the DCD donation is the same or different. Clinically, after people start using the DCD lung, the clinical outcome is compatible with the classical DBD, but are they really the same or not? And also if the EVLP will make a difference on top of the original difference between the DCD or DBD or not, no one knows. So she collected over 177 samples from the DBD lung and the 65 from the DCD lung, then performed the transcriptomic study, basically try to compare if there's some differentially expressed gene. So interestingly, what she found here, the red color means DBD, the green color means DCD. In all samples, the top differentially expressed gene, you can see is clearly separated. And also from the non-EVLP lung or EVLP lung, so they all showed a clear separation. So DCD and the DBD samples are not the same. So this is the first interesting finding we found. But what's the difference? So then she used the pathway analysis, try to understand if there's some differentially expressed pathways. And I apologize for all the word, it's too small, but if you pay attention, you can see cell death, apoptosis, cell death, and so on and so on. So basically, all the DCD pathway enriched in the cell death related mechanism. And the DBD is enriched with the inflammation. So this is a very, very intriguing and very interesting. So this work is published in the European Respiratory Journal. Then we also try to validate that by comparing the gene expression profile in the donor lung. And then we're also using the protein concentration measured from the EVLP perfusate. Then again, we can see the IL-6, which is a very important pro-inflammatory cytokine. It's higher in the DBD, lower in DCD. IL-8, the same. IL-1 beta is the same. So they all show the significant difference, which match the discovery from the gene level as well. So this tells us if we have received a lung from the DBD donors, we should try to treat them with anti-inflammatory drugs if possible. Then if we receive the DCD lung, we can think about to blocking the cell death as a potential therapeutics. So this, to me, is very, very important. So the second work we did is done by a PhD student, Aaron Wang. And Aaron tried to understand the difference between the ischemia-reperfusion process occurred in the lung transplantation setting versus during the EVLP. So he gave the rationale like this. So for the regular lung transplantation, the donor lung will be transplanted to the recipient. But for EVLP, the same donor lung will go through the EVLP perfusate circuit first. There's something in common. For example, they are both performed at the normal thermia, means body temperature, both being ventilated, both being with the circulation. But the big difference is for the regular transplantation, the patient will also receive the blood from the recipient. So this blood-mediated reperfusion is really the trigger for the injury. Then this factor is not exist in the EVLP. So, but overall, there's many similarity. And for all this similarity, would those similarity give us some hints to tell us what will be the ischemia-reperfusion associated signature during the lung transplantation, and which will be the specific signature during the EVLP, and what could be something in common. So that is his experimental design. And I have to say, Aaron is really a genius, and in terms of research, he's a very capable young individual. So what he found is for the transplant specific signal, the biggest signal is inflammation. You can see all these cluster, all the red dots means enriched or upregulated gene sets in after reperfusion. Then the second enriched cluster is cell death. Then the cell metabolism related gene are downregulated. So this is a very, very intriguing. So it's not a surprise to us because we know the inflammation and cell death are the major troublemakers, but we do not know by using this non-biased technology, we can find such a signature exactly like what we would like to see. So this is really amazing to people like me and Shaf. We have done research for so many years. And also the inflammation pathways mainly related to T cell, B cell, and the lymphocyte and the adaptive immunity. Then for the EVLP specific gene sets, the number of differential genes are much less, and also the cluster is much less. But interestingly, most of the gene cluster we discovered in the EVLP is related to a leukocyte function. And more interestingly, they are all downregulated. So this is also, again, it is a surprise to us. And also we found the vascular process genes are enriched. So this tells us why EVLP may have some kind of an improvement in terms for the organ function, because they have a downregulation for the leukocyte-related function. So this may help us to making some argument to promote further study in the EVLP research. But more interestingly, is there's a group of genes shared by both transplant and EVLP. And again, the major signal is inflammation and the cell death and heat stress. The downregulated gene cluster include metabolism and the protein synthesis. So when you look at this, like really, we got very, very excited. That means during the EVLP, the signal related to cell death and the inflammation actually is already being activated. So that suggests perhaps we should try to develop some anti-inflammation, anti-cell death during the EVLP, then to greatly improve the organ function before transplantation. So this is amazing. So it's very intriguing to us. So indeed, we do have some study. For example, we're using a drug called alpha-1 antitrypsin. And this drug has been demonstrated has a anti-inflammation, anti-cell death function. By using that, in the pig lung EVLP or in the human lung EVLP discarded from the clinical program, we see a dramatic improvement of the organ function. So that suggests we should thinking about seriously to apply anti-inflammation, anti-cell death treatment during the EVLP. And also the inflammation pathway is more related to innate immunity. So this study is really amazing, but it did not connect the ischemia-reperfusion process with the clinical outcome. So we do not know, we do know inflammation and cell death are activated, but what we do not know is if this signal is really relevant to patient outcome. So then Christina, together with Jason, is a research assistant in the lab. So two of them using a combined so-called multi-omics approach to further study this problem. Again, the lung was sample was collected from human lung before, after reperfusion. Then the sample was compared. The metabolomic study was conducted by Jason, and then to identify differentially expressed metabolites. Then the gene is analyzed by Christina to find a differentially expressed gene. So then we put those two together, then go through a software called OmicsNet. So this OmicsNet can try to figure out the relationship between the differentially expressed metabolite in the gene. So this is then to get enriched biological pathway. So this is a strategy we used for this particular study. So the results is very complicated. I will only bring you the bottom line. So this is the way how the results from the multi-omics looks like. And I like this very much. For example, this is a 3D, looks very much like a global, and this looks like the busy airport. And this is like the airlines to go to some other smaller airport or some other places. So it's really help us to figure out the connection and the central hub of the signal from the metabolite and its related gene and so on. And again, this is another way, it's a firework. So you can project the related signal from one layer to another to understand the relationship, how this firework is shooted from the bottom. So this is the metabolites, and this is the gene related to this to this metabolites. It's very interesting. Then this is the 2D mapping. So by performing this multi-omics, then we can find the relationship and also top ranked pathway. And the number one, the highest, the top ranked pathway is a purine metabolism. So here, basically we found a gene called PNP, and this protein actually mediates the conversion of this purine metabolites. For example, from guanosine to inosine, and adenosine to inosine, and so on. So all this collectively, you can see a lot of green arrows going down, and means with the consumption of these metabolites, there's an increased production of the final products in this pathway is a urate or uric acid. So this is really interesting. So here, the CIT and the reperfusion, you can see the downregulation, then this is upregulation, the upregulation. So the two major upregulated signal, one is the uric acid, one is the PNP gene. Then most of other metabolites downregulated. Those change really related to clinical outcome. So this is a question we want to answer. So then we analyzed the level of inosine with the clinical outcome. Here is the ICU length of stay or the ventilation time. And we found the lower level of inosine is associated with a longer ICU stay and the ventilation. And on the other hand, the higher uric acid is associated with a prolonged ICU stay. So this tells us the gene expression may provide a hint, but the gene products or the function of the gene or the protein mediated by the gene is really tells us what's going on. So this tells us by using a multi-omics, we will be able to connect the gene, hopefully also with the protein and with a metabolite, then collectively give us a whole picture to understand all this omics change with clinical outcome. So I showed you by using the transcriptomic studies, we can study the gene expression change at tissue level. But we all know the lung, as well as other solid organs, is a very complicated tissue structure with many, many different types of cells. Each cell may respond to the ischemia-reperfusion condition differently, and each cell may play a different role in the organ injury. Then how can we really understand these details? Then we have to thinking about a way to study the mechanism at the single cell resolution. So there's a technology now very popularly known as the single cell RNA sequencing. So this is it's very, very powerful technique. But the experimental design underlying the technology actually is very simple. So here we got a small biopsy from at the end of the cold preservation. Before transplantation, we got another biopsy immediately after transplantation. Then we will be able to see what kind of a signal. For example, in the donor lung, in the CIT, the cells all from the donor. So then you can have a different cell cluster, means from different cells. But from the recipient, in addition to the donor cells, then we also have a recipient cells. Then we need to have a particular software to differentiate those two source of the cells. So then we can further perform analysis, for example, the gene change before, after transplantation, and also to compare the donor and the recipient with the same cell type, but a different source. And we also can study the cell-cell interaction as a pathway. So here we found in the human lung, the macrophage and the type 2 alveolar epithelial cells, type 1 alveolar epithelial cells, and also endocellular cells are in the richest cell population in the lung. And also those have dramatic change in terms of gene expression. So what we found, remember, I showed you in the EVLP perfusate, there's an increased level of IL-1. But now we found actually IL-1 is mainly from alveolar macrophage. Then we also measured IL-8. Now we know IL-8 actually type 2 cells is one of the enriched source for IL-8. For IL-6, it's mainly come from endocellular cells. So that is really nice to know. So when we see a dramatic change of a particular cytokine, now we will have an estimation to know perhaps the activation is mainly reflected endocellular, epithelial, or inflammatory cells. So we also found the infiltrating recipient cells, and we particularly found in the monocytes, the donor and the recipient cells have different gene expression profile and play different role in the inflammation, and so as the T cells. So the detailed results will not be presented. I will only just show you the concept and the experimental design. So we also can do the cell-cell interaction. For example, what we found is for the IL-8, like what I said, it's mainly from macrophage. Then from macrophage, it recruits both the donor and the recipient monocytes and the neutrophils. So this is the donor monocyte and the neutrophils. This is recipient monocytes and the neutrophils. Then here, from type 2 cells, I mentioned it releases IL-8. Then this signal will recruit neutrophils from the recipient and the neutrophils from the donor as well. Here is the endocellular cells release IL-6, then from IL-6, then recruit monocytes from both donor and the recipient, and also further stimulate type 1, type 2 cells. So we think perhaps the endocellular cells is really the initiator for the ischemia-reperfusion injury by triggering signal not only to directly recruit the inflammatory cells, but also activate the epicellular cells, then trigger the further amplify the signal as well. So those results are very interesting. Of course, it's not further validated yet, but it gives us a clue and what could be the next way to go. So in summary, I just quickly go through our preliminary work on the omics studies in the ischemia-reperfusion injury. First of all, we found the DBD and the DCD are different. Secondly, we found the metabolite, purine metabolite could be related to clinical outcome. Of course, we only analyze the one pathway. If we continue to analyze the other pathways, we may find more molecule related to clinical outcome. And inflammation and the cell death are the two common mechanisms between reperfusion and ex vivo lung perfusion. So could it be a beautiful therapeutic target for drug discovery? And also EVLP could be a platform to repair and reconditioning the donor lung. So before I conclude, I just quickly show that this kind of a work is impossible without a tissue bank. So we are fortunate that we have accumulated the tissue banking for over 2 decades. Many, many samples are collected and analyzed, which help us to understand the mechanism for lung injury and also help us to improve the patient outcome. So we have collected many samples, include donor large airway bronchial wash and donor lung tissue biopsy and donor lung tissue collected during the EVLP perfusate and recipient lung tissues and the blood sample after transplantation. So this really gives us an enriched resource for further study. So I hope that in the future, hopefully with the increased research funding, then we will be able to further conduct those kind of study at a larger scale. So before conclude my work, I want to particularly acknowledge the long-term collaboration with the Shaf, with Marcelo, and also with those lab members who performed most of the work I presented today, and the funding sources. Without those support, it's impossible to do this kind of a work. Okay, so I think I will stop sharing the screen. We'll be happy to answer any questions.
Thank you, Dr. Liu, for such a great presentation. Well, now the floor will be open to questions. I'll be happy to moderate those. So I'm going to go from left to right. Excuse me if I'm impolite, but I'm doing my best. So Dr. or Mr. Gunaratram, please go ahead.
Thank you. Dr. Liu, this is very nice work, a lot of work that you presented today. Really enjoyed your talk. You've presented as you identified many different pathways, including metabolic pathways. How do you tease out which ones are more important or less important or actually pathogenic?
It really depends on the objective of our study. I think at this moment, we studied, for example, the purine metabolism pathway. it is the number one highly ranked pathway. So then we did a follow-up with that. But like what I mentioned during my presentation, it's definitely necessary to study other pathways as well. So we should not ignore the smaller signal. But today I was told that we should try to keep the presentation relatively simple and easy to follow. So that's why I did not go through the lot of technical details. But I think so far what we studied is follow the rank of the pathway.
Kathy Bain, please go ahead.
Yeah, if you guys can introduce yourself, let me know who you are, then we'll
Sure.
help
My
you
name's
to-
Katie Bain. I work at the CDTRP. Thanks for giving us so much love during your presentation and for giving such a nice talk. I have a naive question. I am not a basic science expert. You showed a slide that had the different metabolic compounds tied to clinical outcomes over time.
Right.
Is there, this is probably silly and naive, but the ones that are low and that have poor outcomes, is there an option to just give supplements and see how that affects the clinical outcomes over time?
It depends. Even though I mentioned those are metabolite, but actually what I did not mention is that particular pathway is one of the very well-known oxidative stress pathway. So I think that fit our knowledge in the literature very well. So basically, why those metabolites so much related to clinical outcome, because they really related to the oxidative stress, which is the trigger for the inflammation. So so from that point of view, I will imagine we should try to do some antioxidant treatment rather than to do some supplement. For example, inosine level is lower, so shall we give inosine? I think perhaps we should try to blocking the oxidative stress.
What are the mechanisms to do that?
There's many different drugs, different way people can think about how to develop that. Of course, as what I showed in the beginning, we have a drug screening test, right? So then you can thinking about possible drugs or treatment related to this mechanism to collect as many as possible. Then you can go through to screen them to see which one is more effective than the other.
So that could be at future directions.
Right, right.
Oh, great. Thank you so much for answering my question. I really liked your talk.
Thank you.
Erica, please go ahead.
Hi, Dr. Liu. My name is
Erica.
Erica. I'm also a CDTRP member. I have a background in sciences, but very left field in the sense. I have more of a degree in animal neurodevelopment and obstetrics. So my main question is, omics is a field I've never heard of. Is it new or is it actually an old practice?
It's not new, but I can tell you a story. Many years
I'd love
ago,
to hear your story.
one of my colleagues, he's a professor in biochemistry. He got a car plate, and it's a proteomics. It's as a gift, a birthday gift from his children. So he very proud to put it on his car. Wherever he drives, people say, "Oh, you must be a biochemist." But now the scenario changed very much, and people like me, I'm not a biochemist at all. But we are started doing the omics. So omics is not new, but it was a kind of a very, very fundamental, very basic research tool for a while. Now become more and more popular by the clinician scientists like us. So by the way, if any question come to me, it's Erica's fault, and she told me what to do, what I cannot do.
As long as I don't have to answer the questions, we're good.
Thank you for your help.
Thank you.
Francis Mingyao, allez-y. Please, Francis, go ahead.
So thank you for pronouncing my name correctly. So first of all, I'm a research associate in Marie-Josée Bert's laboratory in Montreal. And I'm pretty interested in the endothelial cell death, mostly apoptosis. So it was a really interesting presentation. Thank you. And an incredible amount of work. I have some questions though related to your work. So you've presented results on transcriptomic data. Are you planning or did you study transcriptomic profiling of microRNAs specifically?
No, we did not. But I have to say the data I presented was from the microarray study. However, now, as you know, the so-called next generation sequencing, right? So now instead of doing microarray, people will start doing a non-biased sequencing for all the RNA signals. So in that way, people will be able to collect all these MIA and other smaller RNA molecule, which has never been described in the past. So because of the power of this RNA sequencing, we will be able to capture all this now.
Great. And based on your comment, are you planning to use single cell RNA sequencing to just to follow up question from Lashman, so to identify, for example, the type of cell death pathways that are specific to epithelial cells like type 2 alveolar cells compared to endothelial cells?
That's also a very, very good question. So first of all, the single cell RNA sequencing project is already done, and the paper submitted to a couple of top-notch journals being rejected again. So now it's down to the latter, but now it's more to the realistic journal to be published. So we did that, but that study was with a limited number of samples. We did a 6 pair of donor and the recipient lung. So we basically show the feasibility and the background information of the at the single cell level and what kind of changes it may happen. But in terms of from there to go for the more clinical outcome, we needed to using a very large sample size and to do more study. So I applied the grant to CHR a couple of times. The grant ranking, the score is going up and up, but then this eventually told me, please do not resubmit it. So they are not interested to review it. But I can see the challenge in terms of research funding for all of us, and it's really a major limitation. So we have a lot of great ideas, but in terms of doing that, it's really limited by that. Then in terms to understand the single cell type for cell death, for example, what type of cells will go on under which type of cell death. And we have a cell culture system. Remember, I showed one slide, we have cell culture. So I do see on the list that my former PhD student, Dr. Kim, Hee Kim, is at the meeting. I'm surprised that she's with us. And she actually was the first person did the necroptosis study in the cell culture model. And she identified the lung epithelial cells under the simulated ischemia-reperfusion condition can go on under the necroptosis. Then also she found the necroptosis could be cell death receptor independent. It could be death receptor independent mechanism and mediated through an intracellular oxidative stress. That's the terminology we used. Then we'll also find some particular drugs can block those and then to protect the cells in culture and also protect the animal from the transplantation model. So now my lab actually developed a particular interest to do the cell death study. And recently we put together a review article called Necroptosis in the Organ Transplantation. And through that review, I recognized Dr. Zhu Zhang, and he's also here. And it's really the pioneer for the necroptosis study in the organ transplantation. His team pioneered the kidney and the heart research in the necroptosis, and it's absolutely leading or cutting edge research. So we are so happy to know that for the cell death, I think it will be one of the very important therapeutic target for the organ transplantation, not only for the lung, for all the solid organ. I think for sure it will be. But the complexity is that in addition to apoptosis and necroptosis, there's so many other So ferroptosis, the paraptosis, I even cannot pronounce them properly. So my question is, are they really the different elephant in the room, or they are just the same elephant, but with, we as blinded, we only touch a different part of it. So I wanted to understand that and try to see, shall we develop a cell death specific treatment, or shall we develop a treatment which can target all the different type of cell deaths? So to answer your question, we do need at a single cell level work to understand each cell type under the same ischemia-reperfusion condition, which way they are going to die.
Thank you very much.
Thank you for the question.
Thank you. We have a chat box question from Zion Hong. It says, "Hi, Dr. Liu. Thank you for your important research work. I have one question. What is the most common infection after transplantation among the recipients in Canada?"
I'm not a clinician, so I perhaps is not the best person to answer that. But by talking to my clinical partners, I think the reoccurrence of the CMV infection is a major problem, and also the fungus infection could be a problem because the patient is under immunosuppression. But again, I'm not a clinician. Sorry, I cannot answer that well.
Okay, thank you. We have a question from Sonny Dahnani. I'm sorry, Dahnani. Go ahead, please.
Perfect. Thank you. Can you hear me?
Yeah.
Hi, Sunny Dahnani. I'm an ICU physician and a donation researcher in Ottawa. Again, congratulations on years of work and really making Canada, the leader in this area of transplant research, right? We're also proud of it, really.
I think collectively we are the leader. Canadians are the leader in the organ transplantation.
I think you're right. So this is just such a great example of that.
Yeah.
Coming from the donation side and recognizing that, your end goal seems to be like, medications and drugs that may impact inflammation or cell death. And it's pretty easy to see how you could just give it when you're on EVLP and you're on ex vivo. Do you think we're going to be challenged by giving medications and drugs to a DCD donor, so at the time will be still alive? Do you think there's going to be challenges giving either for research or eventually for therapeutics?
I think I was asked by the Journal of Heart and Lung Transplantation to writing a review article on the brain death, the effect of the brain death to the inflammation. So through preparing that review article, I learned actually donor management is a very, very critical, very important area of research. And I feel perhaps you could be the leader of that. And we do need a very, very good research on the donor management part. So I think this part can never be underestimated. I personally know very little, but I think that area is very, very important to study. But I don't know how, and maybe we can think about how to enhance the donor management part. So I showed one slide about the management of the donor organ. So that was from one of the review articles we put together for cell death in the lung transplantation. Initially, it only have a EVLP part. So I highlight the significance of EVLP. Then the editor and the reviewer say, wait a minute, actually you ignored the donor management. You ignored so many other things. And also not every center has a EVLP access. So you need to think about how your work can give a general guidance to people in terms of management. So I redraw that figure by including the donor management, also included the intra-transplantation management and the post-transplantation management. I think that picture is much more completed, and also it's based on the comments from the reviewer. And also I feel that comment is absolutely important. I really cannot emphasize enough about the donor management. I hope that maybe one day you can give a talk. Then I definitely will be very much interested to listen.
That's a deal. I think engagements like this or CDTRP in general maybe are avenues for collaborating to get the science that you're doing to really then use ICU researchers and a very strong Canadian donation research group on the ICU side and death and dying side, that we could start implementing these sort of drugs and medications and samples in the dying patients. I think that's
another
really,
thing I can
really
comment on is I reviewed one paper. People studied the donor plasma for proteomics. Then they found the relationship between the biomarker with cardiac transplant outcome. But those patients, actually, those donor also contributed to other organ donation as well. So one very important point actually is the data should be also correlated with other organ transplant. So I think the coordination across the different organ system is very important, especially if you have a donor sample, donor plasma sample. That signal can be really utilized by all the different solid organ transplant teams, not only by the one who wrote the paper. So that's the comments I made in my editorial comments on that. I think you guys, if you can have all the samples and we can collaborate, we can work on that and analyze the signal, then shared with all other organ transplant team as well.
Well, thank you very much. It's been a great presentation, Dr. Liu, again. We thank you dearly for the time you spent with us and the great knowledge that you transferred to us.
I really enjoyed it. Thank you very much. Thank you for sharing this.
I see that our time now is just about up. So I want to thank everyone for being here and for the questions and comments throughout today's presentation. Dr. Liu, again, on behalf of CDTRP, I want to thank you. We want to thank you for presenting. And if anyone has additional questions, comments, or suggested topics and speakers, we welcome you to contact Erica K.T. Croft at ekcroft@cdtrp.ca. And again, thank you and enjoy the rest of your day. Have a great one.
Thank you.